She started her academic career at the Catholic University of Mozambique and then did a master's degree at Bunda College in Malawi. She's employed as a common bean breeder for the government of Mozambique at the Mozambican Agricultural Research Institute. As you'll hear about today, common bean is a critically important food crop in Mozambique and in many parts of sub-Saharan Africa. And one of the principal constraints to bean production is abiotic stress, drought, and low fertility soils, exacerbated by the fact that Mozambican farmers normally do not use inputs such as irrigation and fertilizer. So with support from the McKnight Foundation, Anica has been studying how to phenotype root traits, how to cross plants to get better root phenotypes, and then how to do genetic analysis of root traits in this species. So with that, Anica, please.
Thank you, Jonathan, and thank you, everyone. It's my pleasure to be here and be talking about one of my favorite crop and food, beans. First of all, I would like to present my topics that I'll be going through my presentation. The first one is about compare root phenotypes of root traits, or compare root traits in different Phaseolus vulgaris. The second one is about crossing between Andean and Mesoamerican gene pool and try to integrate root fins into Phaseolus vulgaris. The last chapter is about GWAS, Genome-Wide Association study of root traits in Phaseolus vulgaris. Now, talking about beans, that is a major source of protein in the world, especially in countries that cannot or do not have animal protein. Over 200 million of people in Africa, they have beans as staple food, a primary staple food. Now we can see that there's a lot of variety of beans that we find in Africa. So let's see where is the bean production. Most of the places are tropical and subtropical countries. You can know that beans is a tropical culture. And we find them most of in Central America and South America and most of African countries, though the production is very low. Why we having very low production? There is major problem that affect bean production. Most of them, they are biotic stress, drought, and phosphorus deficiency. We can see here in this picture that drought affects 73%. And this is a lot for, especially in countries that have low income for agriculture. Okay, maybe we can think about fertilizer to respond to phosphorus deficiency. But let's look a little bit about the world in general, about the soil fertility in the world. Looking at this map, we can see that most of the countries, they have low soil phosphorus availability. And most of the places that have been production, they have very low soil phosphorus availability. And we know that phosphorus is a major nutrient for bean production. Now, maybe we know that our soils are poor. Maybe we need to think about fertilizer. Do you think that fertilizer is a solution for that? Maybe for some countries, but developing countries, fertilizer is not a solution. As we can see in this map, that most of the countries in Africa, they don't use fertilizer, or the use of fertilizer is very low. We can talk more, we can focus more. For example, in Mozambique, we can see that the use of fertilizer is 0 to 1 kilogram per hectare. And this is nothing. Then talking about Mozambique, because I went to Mozambique, talking about Mozambique, most of agriculture in Mozambique is owned by small farmers. And we know that the soils in Mozambique are poor, they have very low fertility, and most of them, they don't have income to buy fertilizer. For that, we propose to use traits based on selection to develop and release improved cultivars. How are we going to do this? How we are going to address the abiotic stress? First, we need to identify themes related to abiotic stress, integrating them greater themes. And the last point, release adapted and accept cultivar according to the region. I feel like the last point is the most important for, especially for farmers. Okay, we can do the first 2 points in the lab. We can find greater things. We can integrate them, and everything is great. But the time that we are bringing to the farmers, we need to be very careful. I'm talking this because in Mozambique, we have a black bean, and most of the small farmers, they reject the black beans in Mozambique. They say that they cannot eat for dinner because it has a dark color, and the beans is, dinner is dark. Most of the place, they don't have electric, and they cannot see. They don't have light, and they reject the black beans. Although most of the people, or some people, they find that the black beans it has higher value of nutrition value. But the farmers say no. And at this point, we need to be careful when we bring new cultivar for the farmers. So going there, here in this picture, we can show the two main problems that we have in Mozambique: drought and low fertility. In the left picture here, we find that this soil is very dry and the yield is gonna be very low. We can see definitely that the farmer is not going to have anything. But in the right side, we see that the field is low P stress here. And okay, maybe it's going to have something explained as water, but the low fertility soil is going to affect the production. Then, as you know, Mozambique, most of the farmers, they are women. And the men that go there and clean the field, they do the first cleaning, cut the trees, the big trees, open the field, and the rest of the work they give to the women. And we see here that there is one woman here in the left side here. She's here, she has a small baby, and she has to take care of the family, the house. At the end of the day, she has to take care of the field. As a result, she cannot take all the work. And we see our field is complete, full of weeds. In this case, she's going to have very low yield. Although we have problems with low fertility and drought, but if we clean the field, at least we can increase a little bit our yield. In the left and the right side, the women were invited to come to research station, and they have plots there. And the extensionist or the research there help or teach the women how to keep the fields clean and how beans grows, give all the teaching for the women there, because they know that most of them, they're in the field or they work in the field. for them to increase their production. At the end of the day, the men that come there and take their yield and go and sell. The men are responsible to sell all the products, go to the market and sell all the products. So in this video, I'm trying to show a good genotype of beans and how roots behave in the soil and how roots can obtain water and nutrients in the soil. Why I'm saying that this is a good genotype? They have a lot of adventitious roots, a lot of basal roots, and have higher root hair length. At the end, as long tap root, and we can see this soil is dry. And they have all the greater conditions for phosphorus. We know that phosphorus is an immobile nutrient and stays in the top of the soil. Having roots that are shallow roots and a lot of root under the top of the soil, they can achieve or can be greater for phosphorus acquisition. And having long tap root for dry soil is greater because the roots can capture water in the deep of the soil. So I was talking about good and bad genotype. Maybe I need to explain what is bad genotype or what bad genotype looks like and what good genotype. Okay, we know what is good genotype. In the left, we have the left side, we have one genotype with very few adventitious root and basal root. And the basal roots are formed, or most of them, they are in deep angle. While the right side, we have a good genotype that have a lot of basal roots, a lot of adventitious root, and the long tap root. And you find that it has long and dense root hair. And they have good characteristics for phosphorus and the water, for phosphorus capture, or can improve a phosphorus capture, the genotype on the right compared to genotype on the left. This because we find phosphorus in the topsoil. For this genotype, they cannot get a lot of phosphorus in the soil because most of the root goes down deeper, they are deeper roots and go down. While this one, all the roots are in the topsoil here, shallow, and get a lot of phosphorus that find in the topsoil. Then we are talking about basal roots, and we need to know what's a basal root. Basal roots arise from different whorls, and each whorl has 1 to 4 basal roots. And you can see here in this picture that genotype A has 3 whorls here, that each whorl is composed of 4 roots, while genotype B has only 1 whorl. And previous study done in this lab found that the more basal root, the greater will be for soil volume exploration. And wild beans have 1 to 2 basal root whorl number compared to cultivated beans that have more than 2 basal root number. The same study in the basal root, Miguel, a PhD student, was a PhD student in this lab. He did a study about basal root whorl number, and he compared basal root whorl number and we showed the biomass. And he found that the more whorls, the greater is the biomass at low P. So going for the second traits, our second traits, we talk about basal root, and now the second trait for phosphorus capture is root hair length and density. We can see here in this figure, in this picture, it shows long hairs here, root hairs and density, while in the right pictures shows short hair. What is root hair? Root hairs are small tubular that penetrate between soil particles to obtain immobile nutrients and improve the soil rhizosphere that it can enhance the water acquisition. Root hair is involved in nodule formation in legumes and is cheaper and high beneficial to the plants. So Miguel did a study, compared root hairs length and the shoot phosphor content. And he found that there is a higher correlation between root hair length and the shoot phosphor content. So he decided, or he looked into traits, root hair length and basal root growth angle. As a baseline, he had root hair length, short length and deeper angle. So if we change the length, or if we change the growth angle, we have shallow angle and short length, we find an increase of 58%. Then if we change both traits, we have long hair and deeper angle, the increase goes to 90%. If we combine those good traits, long hair and shallow growth angle, we expect to see an increase of 150, but actually we see an increase of 300%. This is because of trait synergism relationship. Now, going for our first chapter, here we have to compare root phenotypes in domesticated and wild species, Phaseolus species. We expect to see significant variation in basal root number, root hair length and density across different Phaseolus species that could be related to their abiotic stress tolerance. Our goal in this study is to identify species that have root fins related to phosphor efficient for breeding. Here we had 31 species that is involved for 51 genotypes. We had in these 51 genotypes, we had 5 domesticated and the 46 wild genotypes. We grew them in a greenhouse and roll-ups. In the roll-ups, we grew them for 14 days, while in the greenhouse, we grow them for 6 to 7 weeks. We sample for basal root hole number, root hair length and density, and the root anatomical. In the lab, we had this is the procedure that we use in the lab. The roll-ups, as I said, we place them in germinating paper. We place 5 seeds and we roll them as cigar. We place the cigars or the roll-ups in the container and we keep them for 4 days in dark chamber. After that, we remove the roll-ups for the light for 10 days. We sample, and we did sample in the basal roots, and we use Toledan blue to help to visualize the root hair using microscope. We took the picture, and using image J, we analyze the root hairs. We measure the length of the root hairs and the density of root hairs. For the greenhouse, the scenario was a little bit different because we had cut the shoot, separate the shoot for the root, and slowly, carefully remove the soil, the media, separate the media from the root, and after that, wash the roots. You can see here there is Camilo here. Camilo is a student here in this lab, and this Sergio was a visitor professor from Brazil, and they helped me washing my roots here in the greenhouse. For the field, in the field, we have a little bit of work, and we use shovel. We can see here in this photo, Jonathan here explained some students how to dig the roots. So we dug the roots, we collected the root, and we take the roots, we wash the roots here. We have containers, we water and wash the root. At the end of the day, we sample the root, and we keep the roots in ethanol, 75%. If we need to do other studies, if we need to do for the root hairs, we need to keep the roots in 75% concentration of alcohol. Then, as a result, here we can see that wild beans differ from domesticated beans in root and shoot biomass. Here, Camilo is trying to measure the roots here and say that, oh, this root has almost my height here. And we see that we have wild beans here, and in the right side, we have domesticated beans. The domesticated beans has very short root, and the biomass or the leaves are completely different than wild beans that have tiny leaves and very few compared to the domesticated beans. Going this, we just try to analyze, to see the variation among the Phaseolus species. And here we find that the domesticated beans have more basal root wall number compared to wild bean. So we did this for root hairs, length and density. And we find that in the root hair length, root hair density, some genotypes have higher density compared to cultivated genotypes. Some wild genotypes had higher length compared to cultivated genotypes, though there are no significant difference here. Going through, we did this for root hair length and density too. Find some genotypes that have higher length. And this brings us, can be a promising results for breeders by selecting wild species with higher density and length, and try in the future to cross and help the farmers, small farmers especially, to allow to have or improve the crop and in poor fields that they have low fertility or drought. So having all these results, we try to find out to know more about the genotypes that we have. And we know that most of wild beans, they are coming from arid soils, dry soil, desert soils, and most of them, they use certain strategies to survive in that environment. Most of them, they have long tap roots or root hairs to see that if it can capture a lot of water or water in the deep soils or some nutrients too. So here in the PCA1 explain 45% of variation, and the PC2 explain 18% of the variation. And we plot here 9 traits. 9 traits in 8 regions. We have Argentina, Bolivia, Colombia, Costa Rica, Guatemala, Mexico, Peru, and USA. This is the place that the seed were collected. We collect the seed in those places. And we try to plot our traits that we had if the places. And we find out that the Mexican genotypes, they are grouped together. Most of Mexican genotypes are grouped together. And this is different than other regions. So we think that they might have different setup of root traits compared to other regions. They might be due to the environment, as we know that most of them, they come from dry soils. And must be the environment that they behave different than other groups. That's why they are separate for the other group. And they have a different setup. As a summary for the first chapter, we find higher variation for root phenotype in Phaseolus species. For fins like basal root number, basal root all number, root hair length, root hair density, and anatomical traits. While the species have fewer basal roots and deeper taproot length, which might be beneficial under stress conditions, especially in water stress. Some wild species present higher root hair length and density, which might be an adaptive response to soil if low phosphorus availability. So breeders can take this advantage in the variation to improve crop cultivars. And improved line will be having healthy water and nutrient acquisition. Now let's talk about our cross between Andean and the Mesoamerican gene pools. We did this cross in Honduras, and we received or they send us the F7 generation that we evaluate here in Penn State or in Rock Spring. So breeders are more focused in improving disease and healed, disease resistant and healed. But they have a lot of opportunity to improve stress or improve resource acquisition efficiency. By improving biotic stress tolerance can be a solution for improving bean production in poor soil and help small farmers to improve their yield and income. And now let's see what is this Andean and Mesoamerican that we are talking here in this chapter. In this map, we can see there are 2 major gene pools. One is Mesoamerican that we find located in Central America, and another one is Andean gene pool that is located in the Andean mountain in South America. And some studies find that the Mesoamerican beans has greater diversity compared to Andean beans. So let's see what is great in this gene pool. What is the contrast between these two gene pools? We find that the Andean gene pool has large seed, valuable seeds, very great seed. Mesoamerican is drought tolerant and disease tolerant. And we see here, we have here some of our parents that we use from Andean that have large seed, and the down is Mesoamerican if small seed. You're going to see more all these parents while I'm discussing or while I'm presenting. And why we need to cross Andean and the Mesoamerican gene pool. We know that Andean has have long and dense root hairs, have greater seed type, and Mesoamerican Mesos have better drought and disease tolerant. And if you combine root hairs and greater seed type, if drought and disease tolerant, would have a positive impact on small older farmers, and they can increase their food and income too. So let's see what we have here for this study. First, we identify the parents and we perform the crosses. We did this in Honduras. We evaluate the progeny from root hair length and density. We did this evaluation here in Penn State or here, yes, here in US. And we estimate the root hair length and density irritability in progeny. Here, we expect that will be segregation of root hair length and density in the progeny. For this study, first, as I said, that we did identify the parents from Andean and Mesoamerican, if contrast phenotypes, and we crossed the parents. We evaluated the progeny under field, greenhouse, and the roll-up. For field and the greenhouse, we exposed them at, we had them at P stress. We had high P and low for greenhouse and field. So those are the parents that we use for cross in your cross. We can see that we have 6 parents for Andean and Mesoamerican gene pools, and 1 parent here just for Mesoamerican gene pool. We can see that they have variation. The Andeans, they have large seed and maybe good color for farmers in Mozambique compared to Mesoamerican. They have dark colors or black colors. But okay, after crossing, maybe we can have different colors or higher variation, the colors. Now we can see that find high variation in this cross, although it's Mesoamerican crosses, and we can see that they have variation in colors. Now we just see variation, phenotype variation for the colors. But after that, we can go more and see if we can find the genotypes variation too. And we can see if the traits that we are looking were transferred from one gene pool to another gene pool. So now what we did is looking at, compare Andean and Mesoamerican gene pools, and we find that the Andean gene pool has greater basal root wall numbers, root hair density, and the root hair length. But one point that brought us here, we find that Mesoamerican has greater taproot length here. You can see that Mesoamerican has greater taproot length. This maybe can bring us in that feeling, that thinking that Mesoamerican, because it's like we find them in Central America, might be in dry soils, and they try to keep growing taproot deeper to have water and other nutrients compared to Andeans that have all the resource available, and they don't have to spend a lot of energy to capture water and nutrients. And after that, what we did here for this, in this study, we did this transgressive segregation. We had to do a selection. We had to select genotypes that performed better under low phosphorus to do further studies. And we did this for root hair length and density. And we find that most of the genotype performed better compared to the parents. In some genotype, they did perform very bad compared to the parents. We did this for, as I said, that we did this for root hair length and density to select them for those traits, because they are traits that we are interested on for phosphorus and maybe who knows drought in some dry soils. And we did the performance. We did another study in the greenhouse. We imposed them to phosphorus, stress, and we find that the genotypes still continue to perform better than the parents. We can see here, I just present only 3 traits: basal root all number, root hair length, and root hair density. And we can see that we are having a promising results that is going to help us to select genotype for further releasing. This is our goal, to release some genotypes in future. After that, we calculate the irritability for root hair length and root hair density under low phosphorus in the stress. And here I'm going to present just irritability for root hair density, that we find that is higher under low phosphorus, except one genotype that performed very well under low and high P has high irritability. One population that performed very well under low phosphorus and IP. So as a summary for this chapter, we made cross between 2 gene pools, was the first point, and we observed genetic variation for the protein in high and low phosphorus. Andean has greater basal root number, root hair density and length, and the seed weight. Mesoamerican has greater taproot length. Some line performed better under low phosphorus compared to the parents. And we observe transgressing segregation for root hair traits and performance under low P. The project continued to outperform parents in follow-up experiment. Now, we are trying here to select best genotype that perform very well under phosphorus stress, because we know that most of farmers, small farmers, they are growing their beans in soil that they have low phosphorus availability. And if we find great genotypes that can perform very well on those soils, will be a good improvement for the small farmers. And the line that perform better under biotic stress, what I was trying to say, can be in the future released as a new cultivar, and they're going to increase income and yield on small farmers. So going for our last chapter, that is GWAS, a study that we're going to do in root traits in Phaseolus vulgaris. Here we have to phenotype 3 panels, Andean diverse panel, Durango diverse panel, and Mesoamerican diverse panel. We had to identify potential genetic control for basal root wall number, root hairs, length, and density. Here, we expect that will be ancestral genes that control root traits common to both gene pool, Andean and Mesoamerican gene pools. Different genes in Andean diverse panel, Durango diverse panel, and the Mesoamerican diverse panel can control root traits. So we had 130 genotype for ADP, Andean diverse panel, 200 for Mesoamerican diverse panel, and 300 for Durango diverse panel. We grew them in the field and the lab, raw labs. And in the field, we had different environment. We have Arizona field, Mozambique, Colombia, and the Washington state. We sampled them for 5 traits: basal root number, basal root wall number, taproot length, root hair density, and root hair length. Now I'm trying to show the fields, the fields that we had. We had the one field in Washington, DC and Arizona, here in the US. We had another field in Colombia, and the last field was in Mozambique. And looking very careful, all these fields, all those fields, we saw that most of them, and they are under drought stress. They are very dry soils. And this might affect our study for the root hairs. We'll see. And here, what we did is look for the variation. And we find that there is higher variation of root hair density within gene pool and between gene pool. This was in the field. And we find a lot of variables such as disease and the soil property that can affect root hairs. And we know that we cannot control field environment. It's difficult to control field environment because most of the time it's dry or it's raining or disease, special for us that we are looking for roots that are under the soil, underground, and it's completely difficult to control all these phenomena or all these problems that we can have. And so what we did, we had roll-ups, and we find the variation for rotate. We did this variation in the roll-ups population for the 3 population, and we find that and the Mesoamerican diverse panel has higher density compared to the rest of the population. And for root hair length, we find that the Andean diverse panel was the best one compared to the rest. After we looked for the variation, we did run the GWAS. We ran the GWAS for the 3 populations and for 5 traits. I just say that basal root number, basal root wall number, root hairs, and taproot length. And here we find high peaks in multiple chromosomes. You can see here, maybe I'll show one of the population, MDP. and we did the GWAS for root hair length in NBDP, and we find high peaks in chromosome 2, chromosome 9, 10, and 11. So having all these results we did for all the traits that we had, and having all those results, we looked more in a single chromosome. Went and looked more in a single chromosome. And we find that in chromosome 11, there's overlapping regions for the three populations, Andean diverse panel, Mesoamerican diverse panel, and Durango diverse panel. And for the 5 traits that we are studying. And now we are trying to see if any of these peaks overlap region or are sharing between traits or gene pool. If regions are sharing between traits, this is interesting because they might tell us some sort of regulator for root traits. But if the regions are shared between gene pools, this will tell us that different populations have similar strategies for root traits and possible nutrients acquisitions. So as a summary, we identified genomes region that breeders can use in marker system selection for root traits to develop lines with better stress tolerance. This region will facilitate discovery of genes that control traits of interest both and within population and between gene pools. As a take-home message, we find higher variation in root traits across different Phaseolus species. We can take advantage on this breeding program for abiotic stress tolerance. Why do species have fewer basal root and deeper taproot? Maybe it's a good strategy for them to have capture water in the deep soils. Some wild species present higher root hair length and density, which might be beneficial under drought stress and low phosphorus availability. Domesticated species produce more root hairs and root hair compared to wild species, possibly because they have greater internal resource. Cross between gene pool can integrate stress tolerant in promising beans. We have identified genomic region that control root traits, and will be compared this between gene pools. This might highlight unique genes and pathways that control root traits. This might be considered between different populations. Breeding for root fins have greater potential to enhance acquisition of soil resource. Could be very important tool for breeders or bean breeding for low phosphorus and drought tolerant in developing countries. In Mozambique, maybe one day I should take all these results and take to Mozambique. So I'd like to acknowledge my advisor, Jonathan Lynch, and all my committee members, Kat Brown, Dave Hoth, Jack Hawthorne, and all the graduates and postdoc, graduate student and postdoc, especially Molly, Hannah, Chris, for the help and support that they give me every day. All the staff, I know that Bob is retiring, but I'll not let him out. He helped me a lot, all on my stay here, all the time that he was here. All the collaborators, from Colombia, North Dakota, Honduras, Zamorano. All my sponsors, McKnight Foundation, USDA, EM Mozambique, and Buffett Foundation. Now is the time that I'm ready to take questions. Thank you so much. Listen.
Thank you, Anica. That was a lot of work you summarized, and very skillfully you occupied the entire seminar period, so there's no time for questions. So the seminar is officially over now. Of course, since we're online, I'd be happy to stay online and take any questions if people have them, but please join me in thanking Anica for a great seminar. There's the little clap icon you can select in Zoom. And once again, I'll stay online in case there's any questions.
Would it be difficult if I could see the summary of the first chapter of the data, please? There was a little bit that I missed on my notes there.
Summary of the first?
Yes, the first summary, or the yeah, the first summary.
Okay, seems that I have to go back like this.
Yeah, sorry about that.
Sorry.
No, it's fine. It was, oh, there it is.
Okay,
There it is.
okay,
There it is.
okay,
Push
okay.
forward. There you go.
Mm-hmm.
Could I get the next one, please? The next slide, not the next slide, the next line. Thank you so much, Anicia. This was a great lecture. Have a great weekend.
Thank you.
Any other questions for Anica?
Hi, this is Liana talking. I was just curious if the GWAS results that you showed, were those specific to the greenhouse environment, or were you showing those GWAS across your 4 different field sites?
I'm showing, okay, the one that I present is from roll-ups, but I'll show or have results for field and roll-ups environments.
Gotcha. And so you don't know yet how consistent those patterns were in the field environments, or do you?
I'm still working on that. I just had from roll-ups. Mm-hmm.
Well, I was just gonna, I just think it'd be really cool to see how consistent that is across all your different environments. That'll be
okay.
Really
okay, I'll share a few when I get the results. Is it okay?
Oh yeah. I really enjoyed the talk. It was really thoughtfully put together.
Thank you.
Any other questions for Anica? Okay, great. Thank you all very much. Thank you, Anica. Enjoy the
Thank you.